Greetings from the ZEISS Microscopy Customer Demo Center here in Dublin, California. Let me begin by thanking all of you for joining us on this inaugural virtual workshop event. We're very excited to have you join us. Your facilitators have put together an action-packed agenda where they'll be presenting emerging technologies, methodologies, and feature-rich functionality that we believe has the potential to advance and accelerate your research. And they'll be doing all of that live from this demo center, which first opened its doors in 2021. And since then, we've played host to in-person demos, remote demos, user trainings, community events, and so much more. And today we're excited to expand our digital engagement with this workshop event. But before we begin, let's introduce your facilitators who are here to help you push your scientific discoveries to new heights.
Hi, my name is Amita Gurur.
Rebecca Duffy.
Nathan Lane.
Chris Bjornson. My name is Sebastian Ortiz.
Darwin.
I joined ZEISS in 1996, shortly after graduating from UCLA.
November of 2020.
On June 5th, 2017.
I joined ZEISS in January in 2017.
January 2020.
December 2019.
My background is in tissue engineering. I studied how to engineer different things like muscle and intestine and lung in a dish. I spent a lot of time in biotech and pharma after grad school, becoming our in-house microscopy expert, before ultimately deciding, instead of continuing to buy stuff from ZEISS, why don't I just go work for ZEISS and play with the toys all the time?
While I was at UCLA, I studied a range of interdisciplinary sciences, from physiological science to neuroscience.
My background for my PhD is cell biology, vesicular trafficking of collagen.
Background has been as a biologist, doing everything from development to neuroscience to stem cell biology. I was in research for a while. I was a core director for a while before I joined ZEISS.
My background is in biomedical engineering and cell biology.
I did my PhD at Hopkins in chemical and biomolecular engineering. I was doing vascular tissue engineering, trying to make blood vessels using biomaterials and progenitor cells.
The fact that I get to help scientists do experiments that they never thought were possible, being able to ask people, what are you trying to do? What are you studying? And learning about fields of study that I would never touch before, and getting to help those scientists see things that really just blow their mind that they never thought that they could do.
Favorite part is coming in and having something new to look at every day and helping people basically get the best images they possibly can to really drive research forward.
Definitely working with new, interesting samples that I would have never worked with before. Like, I had a sea urchin sample at one point. It was really cool to see.
When we're sitting down with a customer on one of our instruments during a demo or training and seeing something live for the first time that they've been trying to do for ages, and being part of that discovery process, sharing in the excitement, because once in a while we get to see data before it's even published, and even for the first time with the researchers.
The opportunity to interact with thought leaders, it's such a privilege to be surrounded by such incredibly bright and motivated individuals. It's motivating for me, it's humbling for me, and it's just a real thrill.
Since day one, I'm surrounded by microscopes all around me. So being at the intersection of microscopy and research, just learning every day how microscopy plays an important part in advancing life sciences research, and continuous learning every day. There's never a dull day.
My favorite instrument is an instrument that adds efficiency to people's lives. I would say it's our 𝗶𝗄cia scan, which is a high-throughput scanner, and there's nothing like being able to let a machine do the work for you.
It would have to be the CD7 with LSM 910. I just like that you can acquire automatically these really nice 3D stacks of organoids in a plate very easily.
I have a soft spot in my heart for the lattice light sheet. When I joined ZEISS, that was the instrument that I was in charge of. It has a very specific application space in terms of long-term live cell imaging, but that is one of those instruments that I've had a ton of those wow moments with customers, and for people that are trying to really push the limits of understanding what actually goes on in a living sample versus in a fixed sample, it really blows everything else out of the water. I still love the confocals, but lattice light sheet is my favorite.
The Lattice Sim, because it's the fastest 3D optical sectioning microscope we have, and because I'm generally impatient and I don't like waiting for things to finish, so I go for the fastest thing I can get my hands on.
My favorite instrument is the Cell Discoverer 7. It's one of those systems where I can use all of the automated features, from what type of sample carrier, automatically setting up my experiment and doing an image analysis. So it's just one of the instruments that I absolutely love.
Right now, it's always been the confocal. So whether it's Spectral, whether it's Scan, now with Lightfield, that definitely is so much power to play with that I really enjoy working on that system.
I would say an eagle. I want us to soar high.
I think it would definitely have to be something white and blue. So I was thinking, what did I think? Like the Arctic fox would be a good one, or a polar bear. I did have a user ask me what could be a good Pokemon name for an Axioscan recently, because they name all their instruments after Pokemon. And I thought 𝗁𝗁𝗶 would be nice. So I think now there's an Axioscan out there named 𝗁𝗁𝗶. So that could also be cute.
I think a group mascot, it might be fun if we had a tardigrade. They're very flexible, adaptable little creatures, and also they're really fun to look at on a microscope. So I think for a team mascot, a tardigrade might be a good choice.
In the spirit of the 990, it would be a platypus, I would say, because it combines all kinds of special forces into one compact little animal. Everything from a bill to venomous spines to a beaver tail.
If we had a team mascot for the Digital Customer Engagement Center, that mascot would be a digital tool belt. That would enable our lives and your lives to be more efficient.
I would pick 𝗶𝗁𝗁. Individually, we all have our unique strengths, but then when we all come together collectively, you can produce some great honey.
Now that you've met our team, we'd like to show you how you could prepare for a virtual demonstration in the future, and how to have the best workshop experience today. Welcome! We're excited to join you for a virtual microscope demonstration. To ensure you get the most out of this experience, we've prepared some quick setup recommendations so you can avoid seeing this and experience this instead. First, let's talk about your internet connection. For the smoothest experience, we recommend using a wired Ethernet connection when possible to minimize latency and connection drops. You'll want at least 4 megabits per second download speed for optimal video quality. If you're using Wi-Fi, position yourself close to your router and minimize other devices using the network during the demo. Next, your display setup is crucial. The microscope software is designed for high-resolution viewing, so use your largest available monitor. 32 inches or larger is ideal. HD or 4K resolution will help you appreciate the fine structural details. Clear audio is critical for understanding the technical details. We recommend headphones or a high-quality speakerphone. Test your microphone if you plan to ask questions verbally, and keep the Teams chat open as an alternative way to communicate. Now let's create the ideal viewing environment. Choose a quiet space like a conference room or private office. Here's a key tip: dim or darken the room. This will help you see the vibrant colors and fine structures more clearly. Remember, this is an interactive experience. We encourage you to ask questions anytime using chat, raising your virtual hand, or speaking up. Request specific views or areas of interest, and share your observations. Your fresh perspective adds real value. Our demo facilitators are microscopy experts who love sharing their knowledge and are prepared to customize the experience based on your interests and questions. If you have any questions before we begin, feel free to reach out to our team. Happy exploring!
Hi, my name is Rebecca Duffy, and I'm a specialist here on our Digital Customer Engagement Center team located out of the ZMCC in Dublin, California. And I'm really excited to talk to you all today about ZEISS's new Lightfield 4D technology and give you a lightning talk on what that entails. This image of the sea urchin tumbling in the background in real time is really an example of how Lightfield 4D is such an advancement in live cell imaging. And I'm going to talk to you a little bit more about how that actually works. And so we've always known that life happens in 3D. And as cell culture methods and imaging techniques develop, our ability to capture these moments has only become more profound and more necessary to capture these 3D processes for live cell or even fixed imaging techniques. Traditional optical sectioning techniques have included things like point scanning on things like confocals using a pinhole to get rid of out-of-focus light. And you can even line scan on confocal as well. But of course, these are slower techniques than, say, something like a light sheet, where we are imaging one sheet or one plane at a time. In contrast, the Lightfield 4D micro lens array allows us to capture an entire volume of an image in a single snap. And of course, what this does is it makes it really, really fast. And what we can see here is an example of a zebrafish beating heart. And so now we're not talking about frames per second, we're talking about volumes per second. And again, this Lightfield 4D allows us to capture something like a fast process like the zebrafish heartbeat, which is acquired in this example at 80 volumes per second. Acquiring this type of image in the past had actually required acquiring multiple Z-stacks at a time and reconstituting all of those images and post-processing when you're analyzing the data. But here we don't have to do all of that reconstruction anymore. We can just capture everything, look at it in Organoids Pro, and track these individual cells in 4D. Again, previously the only way to do this type of imaging was to reconstruct and realign multiple volumes that have been captured over time, which of course is not reality. And now Lightfield 4D really enables us to actually see live in real time what's going on in a beating zebrafish embryo heart. Okay. So this high-speed volume acquisition is not only useful for following fast or long-term developmental processes, it is also high throughput. All right, when we can go fast, that means that we can capture data from many thicker samples in 3D using Lightfield 4D. So for things like spheroids and organoids, this really cuts down on the imaging time that it would take you to image something like a 𝗦ci well or 384 well plate, or really just cuts down on the amount of time it takes you to image one sample. But again, as we're thinking about screening these things in these higher image formats for things like drug screening, you can imagine how much faster it is to get to the image analysis now that you can snap and capture this entire volume immediately. And I'm actually going to have a separate talk about screening a 384 well plate of spheroids later during this workshop. And finally, because we're capturing an entire volume with a single light exposure, it is gentle. Gentle imaging over really long periods of time is important to ensure that living processes that we're looking at aren't influenced by phototoxic effects. In this example, we're following developmental processes during zebrafish ear development over the course of 16 hours. Here we're snapping a volume every 2 minutes. And this is really a great leap forward compared with some of the conventional imaging methods that these scientists studying this have used prior. Traditionally, they've been acquiring volumetric Z-stacks at 5 minutes every acquisition. And during that full 5-minute acquisition, there's deviations during the development processes that you're able to see, because during that volume acquisition, relative to the actual dynamics that are happening, you're capturing that in real time. So you're missing out on some information as well as not getting the full snap instantaneously. But with Lightfield, of course, we can now snap this instantaneously, so you can actually look at multiple animals and at multiple time points and still have a higher frequency that you're imaging at compared to previous techniques. So how does all of this work? That is really what we're going to talk about today. Combined with our new Voluma wide field light source, which is an LED light source, we were able to excite fluorescent samples, send that resulting signal through a microlens array, and that is placed conjugate to the back aperture just before it reaches the camera. And so we're able to snap all of those images at once and transform that information into a 3D snap. Okay, so that is positioned in the beam path conjugate to the back aperture. And so that's what creates multiple images of the same sample. So we're actually capturing 37 different perspectives in the same sample or what we call parallax views. Okay, and so you can see these 37 different parallax views here from the 37 individual lens elements, and each of these views provides both spatial and angular or directional information. And then, of course, deconvolution-based processing considers each of these views to generate a volume that is transformed into a familiar Z-stack. That resulting Z-stack can be used and analyzed as you would any other microscopy Z-stack. So here we're going to talk about how the 37 views collected by the camera chip captures 3D and hence volume information. So we're starting with the graphic on the left. We can see a sample within the Z range that can be reconstructed. And to the right, we're going to look at a cross-section of the multi-lens array lenses. So here we see a line of the 37 lenses, and we've called them 1, 4, and 7. Those are going to be the examples we're looking at. Let's have a look at a specific structure within the sample, depicted here in cyan. On the camera, this structure will be at the same position because it's in the focal plane of the objective lens and in the middle of the Z-stack. Now, if we add another structure in the same plane here, in yellow, we see that it behaves normally. As these structures are not in the same spot, they will be depicted with the displacement we would expect in a wide field image. Okay, so nothing new so far. But now, let's say we have a structure at a different Z position, not just a different X position. Here in magenta, this information will be shifted in microlens 1 and microlens 7. Because it is just below the cyan structure, these two signals add up and are at the same chip position. The mix from the magenta and cyan shown in white here. Because of the displacement of the different views, the Z position can then be determined. And that's a quick snapshot of how that microlens array works. The volume acquired depends on the objective used. So on a 10x objective, you can acquire approximately 3 by 3 millimeters in a single snapshot. And of course, with increasing magnifications, the volume acquired is reduced, but the resolution improves up to a maximum resolution of 2.2 microns in X and Y and 2.8 microns in Z. So that's more than sufficient for cellular resolution on your larger dynamic 3D samples. And of course, starting to work with the new imaging technology might seem intimidating, but there really is no learning curve to Lightfield 4D. We've integrated it into the LSM 910 and the 990, and both of those are workhorses of either individual labs or imaging facilities. And we've ensured that we have things like smart software tools like Smart Setup or Copilot. You can very easily see the volume definition, which we have this really nice infometric from magenta to white to orange, showing you if you are in the top, middle, or bottom of your sample. And of course, the processing that I mentioned earlier, you can process 100 volumes within a minute, but you can also preview your processing and what it looks like by using the sliders in the software to make sure that you are capturing the information that is representative and you're not over-processing your images as well. So it is very easy to use. You just click a couple of buttons and you are imaging your 3D samples very quickly. So the LSM 910 and 990 systems provide really flexible imaging options that allow you to now pick the right tool for the job. Lightfield is the new addition to the platforms, which already offer ARIScan for sensitive super resolution or measuring molecular dynamics, and confocal imaging designed to handle multiplex fluorescence panels. So now Lightfield 4D is an additional tool you can add to your tool belt to further extend the flexibility and Swiss Army knife-like properties of your LSM systems. And of course, it can be added to Zen just like any other track, with streamlined options that focus on the hardware that really matters to you. So for example, if you want to peek behind the veil, you can see the raw data collected as well, because the camera sees the volume and our eyes don't yet. So you can actually go through and look at the sample here. We're going to look at different structures. You can look at all the different structures on the array. You can see which lens we're looking at is highlighted in yellow. And of course, you can actually look at the different layers here. As I mentioned earlier, there's color coding so that you can see the magenta in the bottom, white is the middle, and orange is the top. And of course, you can also preview your processing, as I mentioned earlier. So I want to show you just one quick example. Here we're looking at the neuronal activity in the zebrafish brain that was captured with 10 volumes per second using Lightfield. And here we're following the intensity changes of GCAM. So the gray spheres here highlight neurons that fire simultaneously at very different locations and Z positions throughout the brain. Capturing these synchronous events wouldn't be possible in the sequential acquisition of a Z-stack. You would only be able to take a plane at a time. And then after the neuronal activity was recorded, a Z-stack with ARIScan multiplex was acquired to look at a super resolution Z-stack of the neuronal structure. So we were able to combine here the physiological fast activities in GCamp and then structural measurements in higher resolution with the ARIScan detector, all in a single instrument. So we don't have to move the sample, it's stable environmentally, and we're able to capture a lot of different data on one singular system. And just finally, for further analysis, you can always use end-to-end pipelines developed in ORIBIS. And so the ZEISS ORIBIS Pro software is your modular imaging analysis software. So anything that you're doing from multi-channel 2D, 3D, or 4D images of virtually unlimited size, it's very good at managing huge data sets. And with ZEISS ORIBIS Pro, which was formerly Vision 4D, it's easy to create automated end-to-end pipelines with just a few clicks. So you can use predefined pipelines for common use cases or create your own pipelines using really flexible click-and-play interfaces. But for anything from developmental biology and looking at cell tracking and morphological tracking over time, there's very specific neuroscience pipelines. There's high content screening, which you'll get a snapshot of during the Organoid 𝗶𝗶𝗶𝗀𝘀 talk that I'll give later. And of course, cell biology, anything that you need in the life science space, ORIBIS is going to have a solution for you, whether it is built-in modules that we have, machine learning, or a customized modular click and play. Okay? And that's really going to help you actually make data out of these amazing images you're going to get on the Lightfield 4D. That is our lightning talk for Lightfield 4D. And of course, if you have any questions, feel free to shoot us an email. Thanks so much. All right, I hope you guys enjoyed that lightning talk on Lightfield 4D. As you can see, I've had a little bit of an outfit change since giving that talk, but really excited to show you guys some more cool things with the Lightfield 4D today. If you have any questions, please feel free to email us at the email that is sitting in the top corner here. We also have it in the information below. We're just going to take a really quick break before we jump into some real life applications for the Lightfield 4D and some marine organisms. So just hold tight. We'll be back in a couple of minutes at 10:30. Okay, welcome back. I'm really excited to introduce our next speaker. So we have Dr. Geert de Vrijde. He is one of our application sales specialists. He covers specifically the Southern California region, and he has been going to workshops all around the area, showing off the Lightfield 4D. So just to give you some background, he has his PhD in molecular and cell biology from UC Berkeley. He was in David Builder's lab, and he has used confocal and wide field microscopy extensively when he was doing research to investigate novel mechanisms of epithelial ligand receptor interactions, cell signaling, and tumor suppression function in Drosophila. But his enthusiasm for microscopy actually began back at Utrecht University in the Netherlands, and that's where he did his undergraduate and master studies in cancer genomics and developmental biology. And he did a lot of work at C. elegans cell division there. So Geert is actually in the field at a workshop live right now. We're really excited to actually have somebody basically reporting to us from the field, showing you guys real customer images of living marine organisms and how researchers are now starting to use the Lightfield 4D to uncover things they never thought were possible before. So without further ado, I will pass it to Geert, who is down in Southern California with some really cool applications.
Thank you for that great introduction. And good morning, everyone. My name is Geert de Vrijde. I'm a 3D imaging specialist at ZEISS. I'll be talking today about a recent workshop that we did where we collected some exciting data. We were imaging marine organisms, or actually live imaging marine organisms, using the Lightfield 4D technology that we just introduced. The goal here is really giving you an idea of real-world applications that the Lightfield 4D can be used for. So this workshop was at the Scripps Institution of Oceanography. As you can kind of see in this video here, at Scripps or SIO, the researchers really do interdisciplinary research studying ocean biology and ecosystems. So basically looking at things like the effects of climate change and pollution on biodiversity, as well as working on new technology to deal with ocean-related challenges. Here, you can actually see the building where a lot of the labs that we worked with for this workshop are located. So you can see this is definitely not a bad place to do your science, right next to the beach and right next to where your model organisms live. So it's a great place to do science. Now, as you can imagine, at SIO, there's the labs studying a lot of different marine organisms. So this can range from sea snails, different types of fish, people studying coral and coral growth, different sea urchins, and even sharks. There's a lot of really cool, interesting science and a lot of research that involves live imaging of developing organisms. Now, I'm not going to talk about all the samples that we put on our Lightfield 4D system while we were at SIO, because I could basically go on for hours. But what I really want to do here is highlight some of the really cool applications that we did and also give you some inspiration, right, for if you're doing this kind of research or thinking about like would your research work well for Lightfield. So just to give you a good taste of what works really well. So one of the labs that we worked with was the Amro Hamdoon lab. This here is Evan in the Hamdoon lab, and he's studying sea urchins. So the lab works on sea urchins. So if you've forgotten one sea urchin look like, you can see them in the images here. They're really the reason why when you go to the beach that some people in certain places, you don't want to step into the water bare feet because they're very spiky and live just under the surface. So as you can see in these images, they're actually very mobile. They're not just sitting there, right? Like they're actually moving around on the bottom of the ocean. This will actually become important later in this talk. But what I want to highlight is People often think of sea urchins as these spiky balls that we see here, but that's really their adult stage. They go through a full life cycle that you see mapped out on the right. A big part of that life cycle, when they're in their embryonic stages and later in larval stages, they are actually free swimming in the water. They actually have a metamorphosis stage where they turn into the sea urchin adults eventually that people are more familiar with. So these are the different stages also that the labs work with. And here I want to particularly highlight the juvenile stage that we'll show images of and the embryonic stage of the sea urchin. So those will be the images that we'll be looking at in this presentation. But what the lab is really doing is trying to develop different genetic tools and using screening to study these cell type specific vulnerabilities to different environmental factors or toxins. So they're trying to see with different genetic markers what kind of effect these have on specific parts of the animal. And the way they do it is they develop different genetic approaches to express fluorescent proteins in the animal. So for example, you make a construct that expresses either GFP or RFP in particular parts of the animal. And usually when you generate these animals, you'll get this patchy expression like you see in the drawing right here, right? Like we see GFP just in particular parts of the animal. And so one of the things that the lab really wants to do is screen these animals and then find out in what parts we get the expression, right? Like we want this in the tissues of interest. The animals can be used for either further study to look at those specific tissues or propagate the animal so that you can get a stable genetic line that has fluorophore expression in these areas that we want. So if we take one of these animals, right, like we put the sea urchins in a dish and they're walking around, they're still alive, and we're trying to figure out where the different markers are expressing. What the lab currently does is using a point scanning confocal. And of course, if you're taking an image of an animal in a dish, it's going to take a while if you try to take a Z-stack to get 3D information to collect that data. And of course, what's going to happen if these animals are alive is they're going to move out of that area that you're scanning. So generally, this is too slow, right? Like you can't get that 3D information because the animal's moving around. Now, the only way to get the information at this point is either killing the animal, which is obviously not preferred, or somehow restricting it from moving, which is always going to disturb it in some way. So this is a really interesting situation to bring in light field technology, because in this case, if we want that 3D information of these animals, we can just take an entire volume in one snap. So it would be a really good way to figure out how this looks in 3D without having that problem of taking so much time to get the data. So this is really where we wanted to try out the light field. This is an example of that data that we collected with the light field. As you can see here, the animal is really moving around. This is on a 20x objective, collecting about 50 volumes a second for about 4 seconds total. You can already see in those 4 seconds, the animal is wiggling around a lot. You can see the different spikes. Here, you can even see this appendage where the animal is attached to the cover slip. And then down here at the bottom, you can really see the mouth opening and closing as it's moving around. But what you can really appreciate here too is these patchy areas on the animal where the fluorescent signal is lighting up. So this is actually great. This is ideal. We can see where in the animal we get this expression, even though it's moving. And this is really where Evan jumped up and said, like, I've never seen this look this good before, right? Because normally you wouldn't be able to take this data. So this is an excellent, excellent application for light field, because we can actually see something and resolve something that wasn't visible before. And of course, what we're looking at here is a maximum intensity projection, but this is 3D data. So just to show you kind of what that looks like with a 3D render, we have the 3D information, so we can move around and figure out our angles and look at the different tissues in the animal that are lighting up in 3D. So this really shows that power of using the light field to capture in a short amount of time. Even though the animal is alive and moving, we can still grab this information and easily screen what animals we want to use for any further experiments. Now, that was not the only type of sample that we imaged with the sea urchins. As I mentioned, there's different stages in the life cycle of the sea urchin. So there's also these embryonic stages that you see at the top here, and here at the left you see some of these individual stages. So when the sea urchins are in their embryonic stage, they are actually free swimming, right? Like they're in the water and they're drifting around or swimming around, and it's hard to capture any of these animals, even on any microscope system, because it's constantly moving, and it's hard to set a volume, because even then it's moving in and out of the space that they're into. So we collected a number of these embryos. So kind of this stage that we're seeing down here, the gastrula stage of the sea urchin embryo. We put them on a 384 well plate. We filled a couple of these wells with animals, then brought the liquid down to concentrate them in that volume, but they're swimming free in this volume, and then try to capture this on the light field, because again, this is something normally you wouldn't be able to do on any other system. So that is the video that we're seeing here. You can actually see the different embryos swimming through the volume that we're collecting. This is on the 40x objective, right? It has a 360 by 360 by 100 micron volume, fixed volume that we're collecting, and here we're collecting 80 volumes a second for about 5 seconds. So you can already tell that within just a second this floats in and out of the field of view. So normally you wouldn't be able to get any 3D information, but here we can easily capture that. You can appreciate the different nuclei that are showing up here of the animal, but you can also see there's some rotation happening as they are swimming. Showing it here in a more of a 3D render where you can see that a little better, where they spin around in 3D. So they have a particular way of swimming. There can even be like a chirality to this swimming in the volume. So again, the light field lets you see this volume, capture that volume in that really short amount of time, showing really things that you normally wouldn't be able to see. So again, this is a wonderful application for applying the light field. So just to quickly summarize, right, like these two different stages of the sea urchins, right, like really where the light field 4D gives us a benefit, like it really lets us acquire that 4D data that wasn't possible before. Anything that moves too fast with other techniques, here that's not a problem at all. It really lets us visualize and give us that 3D information even while the animals are either moving themselves or just in a free liquid, which makes them highly mobile. So this is again wonderful to use the light field 4D for. Now I want to switch gears a little bit and talk about a different group that we work with. This is Stephanie Neal in the Lions Lab at SIO, and this lab works on sea snails, specifically embryonic development in sea snails. So here you see a couple pictures of the sea snail type that they're working with, but specifically they're aiming to image the early development, so the early stages where the cells are dividing in the embryo. And how they normally capture this is what they do is they put a number of these embryos in a dish and then image them over an extended time, so about 12 hours, like an overnight data collection, to capture all the cell divisions happening in that time. And then you collect the data every 8 minutes or so. You take a time point. Of course, when you're doing that as they currently do with a confocal system, obviously it takes time to get like a single confocal Z stack, right? Like we're scanning through every individual slice if we're using a point scanning confocal system, and it takes time to get a full embryo in 3D. So it really limits you in how much you can capture within your time points, right? Like you have 8 minutes to collect the data you need, and then you have to get to the next time point. The other factor there is the laser light exposure, right? Like we're having, when we take these Z-stacks on the confocal, we're constantly having laser light hit the sample. What that means is we're always illuminating a very delicate developmental process, and obviously embryos are not happy to be zapped with a laser constantly. So this can definitely affect the development. So we want to figure out a way to do this more friendly. So again, let's try out the Lightfield, right? Let's see if we can do this with a Lightfield 4D and see what we can capture. So here is the video I also showed on my opening slide. These embryos, right? Like they're like a group. There's a group of embryos on a dish. They have two colors, as you can see here. So there's a marker for the membrane and there's a marker for the nuclei. And then, as I mentioned, we grab them like every 8 minutes. We grab a time points, and we do that for 12 hours. So you can really follow all these cell divisions, as you can nicely appreciate here. And this is on the 20x objective on the system. Now, this is really nice. We can see the cells dividing. We see the embryo is happy. But of course, we're collecting a full 3D data set in basically a matter of milliseconds versus something that takes minutes. So while normally Stephanie could be limited by the amount of samples that she can collect in 8 minutes, for example, if a Z-stack takes just over a minute, you can only grab like 6 to 7 animals. Now she had 17 animals on the dish, so we decided, well, what if we just collect more, right? Like we can obviously do this in a short amount of time. Let's see if we can get this animation to go. Let's see. I'll just reset it. There we go. So we can collect a lot more embryos this way. So just how all these cells are dividing, right? If we're grabbing more and more embryos, we just grab all of them on the dish. And we're basically expanding the amount of data that we're getting. We're not limited by the time it takes to acquire the data, and we can get a lot more within that time. And we actually collected a total of 17 embryos, 17, which means 17 dual color volumes every 8 minutes. The other big advantage of this is that you can look through your samples and basically say like, oh, for example, this one at the bottom, we see that one of them was already unhappy at the start. So that's someone or some embryo that we can leave out of the analysis. Or if there's like a angle of the sample that is not as good, we can always omit those, or we can pick out the ones that work really well. Another big advantage of this is you can grab every sample, so any unique event that is happening, you can make sure that this is captured, right? Like we're not limited by only picking a couple of samples that we want to follow through time. So this really allows for a lot more data capture. And of course, when we're collecting this, we're getting 3D data, right? Every single snap provides 3D information. So you can see right here, if we look at the 3D render of this embryo, you can see that it keeps dividing, and you can actually modify that angle and see the developmental process under those different angles. So it really boosts the amount of data that you can get, as well as being able to be a lot more gentle with the developmental process, right? Like we're not zapping it with a laser constantly. So again, a really, really good application for Lightfield. It's a different time scale, right? It's not something that moves really rapidly, but it can still really benefit from using this technology. So here, just to sum up what we've talked about today, right? Like Lightfield 4D is really ideal for these applications for a number of reasons, right? Like we can obviously capture 4D data at like a really high speed. So if our animals are moving a lot, or if there's like any dynamic processes going on, we can easily follow that. Then of course, the short exposures really mean that the phototoxicity is minimized. And this is really ideal for these developmental processes. So we can make sure that everything keeps dividing and keeps going on, and we're closer to really capturing the process as natural as we can, and we don't disturb it by using high amounts of photons hitting our sample. And of course, even just a single snap gives us 3D data. So the amount of information we get in those short exposures does give us like full 3D information. So we can get a lot more data in a shorter time. And again, if we're getting that data really fast, it means that we can always add more points and more samples, and we're not limited by the acquisition time, which is often the case for when we're doing like a focal point scanning approach. So with that, yeah, I'd like to thank you for your attention. I really hope that this inspired you as well to try your samples on the Lightfield, right? Like, it can really open new and exciting ways to get data and answer questions that you didn't think were possible before. Thanks again, and back to our studio in Dublin.
Thank you so much, 𝗏𝗏, for showing us all the amazing data that you've been collecting down at Scripps. And thank you to our customers down there who allowed us to share that with all of you. We are going to take a short break, and when we come back, I'm going to show you all how we can use the Lightfield 4D to very quickly screen a 384 well organoid plate. All right, see you shortly.
Welcome back from our short break. I'm excited to introduce our next speaker. Her name is Rebecca Duffy, and she'll be speaking with you in just a moment. But before that, we heard from her earlier in our testimonials. Nevertheless, allow me the honor to share a little bit of her story where she earned her degree in biomedical engineering at Carnegie from Virginia Commonwealth University to Carnegie Mellon to MIT to industry and eventually landing at ZEISS. We're very lucky to have her in this role, and we hope you enjoy this next session, high throughput screening of organoids using Lightfield 4D.
Thank you so much for that wonderful introduction, Nate. And yes, I'm really excited to bring this next talk to you guys. Give me 1 second while I queue up. I do have a couple of slides before we launch into some live workflows. Okay, so really excited to talk to you guys about this today because I feel like I do bring a little bit of this context with my background, having come from industry before joining ZEISS. You know, I definitely understand that there's often a compromise that you all are making in the biotech and pharma world where you want to be able to get your data as fast as possible. You want that data to be as accurate as possible, but oftentimes time is of the essence. And when you're doing something like screening a 384 well plate, right, oftentimes there's a compromise where you're not going to be able to always capture that 3D data as quickly as you want, and so oftentimes you're having to either just make a sacrifice and take a couple of slices in 2D and try to gather data from that and hope the conclusions you're drawing are accurate. But what I'm really excited to show you today is how Lightfield 4D really enables you to stop compromising and actually be able to get all of your data in 3D if you're doing something like testing drug screening on an organoid plate or a spheroid plate, and actually get all of that 3D data out, port it into 𝗶, our image analysis software, and again, not have to make any compromises and trade-offs just for the sake of time. Okay, so what I have here that I'm going to show you guys today is a 384 well plate. This is a co-culture provided so kindly to us by Inspiro. There's some collaborators that we have. You should definitely check out their website as well. They make plates for you guys so you don't have to always, you know, do all of the 3D culturing yourself, so please check them out. But they provided us this plate so we can practice workflows and see what is going to work for you guys. This is a co-culture of HCT116 cells and fibroblasts in green and in red respectively. And so we have these spheroids here. They're also counterstained with DAPI. So normally if you're trying to screen this in 3D and you're doing a 3-color screen for green, red, and blue, right, you're typically going to probably only image a couple of slices in the middle and hope that your data is representative of the whole spheroid. But now with Lightfield, we're not going to have to do that. And I'm going to use something in Zen, our microscope software, that we call guided acquisition, where basically once you have your experiment setups for your low mag screening, your high mag capture that you want to get in 3D, you're going to be able to just plug all of that in and click one button and get your data out, which again is really exciting when you're used to having to sometimes sit and parse through a plate this big can be painful, right? So this makes it pain-free. So what that workflow looks like is, again, we're going to do a 2D low mag screen first. So I'm going to take an image throughout this whole plate. In this case, we are just doing 2D because the goal here is just to identify where the organoids are in each of these wells, okay? So it's the equivalent of a 5x that I'm going to use. I actually use our 10x objective and zoom it out so that we get a bigger field of view, but it's the equivalent of what you would get on a 5x objective. We're going to analyze that. So what we're going to do is analyze that 2D image and find where the organoid or spheroid is in 2D and quickly find those XY coordinates. And from that table, we're going to store that info and then switch over to our high mag objective so we can actually capture in one snap that 3D info in 3 colors. So if you missed the lightning talk that I gave earlier today, I'll also be giving it tomorrow. This will be recorded. Please check that out so that you can understand a little bit better how we can capture 3D information in a snap. But that is what we're going to do in this workflow. So again, 2D screening, go back and find where exactly the organoids are and have that higher mag objective with the higher numerical aperture so that we can get 3D data and segment out those cells. From there, we're going to segment out the organoids, segment out the red and the green cells, and now we're going to be able to get information from those two populations. Because, right, we have a 384 well plate. That's a lot of conditions. We have multiple drug treatment conditions. We have multiple co-culture conditions. And what we actually want to get at the end of the day, while, of course, I like to get a pretty picture, what we really care about is the data. We really want to be able to draw scientific conclusions from the plate and, you know, give that to your boss and figure out what the next steps are in your experiments. And so we can do that with a 𝗶vis, and we're actually going to use a 𝗶vis hub here, and it's really amazing how quickly we can plug all these things together. All right, so without further ado, I want to show you. This is our LSM-910 down in the lab, right? And if you have seen our confocals before, this will feel very familiar, but now we have a new detector added on it. So on the left side over here, you can see our incubation setup, right? So if you are doing any live cell, we've got all that stacked over here, CO2, heating, humidity. The three detector setup is right here, so we have 2 gas detectors plus the area scan. But what's really exciting is right here, we have our Lightfield 4D, and this is our new detector again that's got that multi-lens array in front of a camera, so that in a snap you get 37 different parallax views that we can combine to a 3D view all at once. And of course, I will just brag a little bit that, of course, in our demo lab, we have all of the toys over here. So we're going to use a hardware autofocus that is part of this stack over here. But this is all a modular setup. So if you haven't used one of these before, these are custom built, and you can pick and choose what you need for your setup and just stack all the pieces together, right? So today, again, I'm going to put a 384 well plate on here. We're going to zoom into the stage, and I'll show you the sample that we're actually working with. This is a simplified incubation setup here, so I'm just going to move the transmitter light arm back, and then I'm going to add our plate. That again, we're looking today at spheroids that are a co-culture of GFP and RFP cells provided to us by Inspiro, and we're going to screen that whole plate. Okay, so we're just going to lock this into the stage, and then I'm going to port us over to the software, since most of the microscope driving is actually done in the software these days. Okay. All right, and so I mentioned we're doing guided acquisition, and so the first step again is going to screen the whole plate. We want to find a 2D image in every single well, and then we're going to pull the XY coordinates where it finds an organoid in each of those wells, right? We're going to let the software do that for us. So the first thing I'm going to do, I did calibrate the plate beforehand. It's just a very quick couple of steps that we do, but now we have an underlying sample carrier. So that means that I can just highlight all the wells that I want to image. If you only needed to image half your plate, you would just highlight half your wells. And then I'm going to hit add position, and it's going to add a position so that it knows that it has to go to every single one of these little yellow crosshairs that are here in the software. So that's step one. And again, I mentioned we're doing a 2D screen here. The way that I set this up for the way that my LSM-910 is configured, I wanted to basically use a wide field equivalent image. So in this case, I open my pinhole all the way up, actually, so we're not optically sectioning. I'm just going to open that all the way up. All I care about is figuring out where that big old organoid is in each well. So we just need to do it as quick as possible of a scan. So we're actually going to drop down the pixel density, because we don't need high mag here. We don't need resolution. We just need to see where is the organoid as quickly as possible, right? So that's what we're going to do. So if you look at the settings that I have configured here, that's what you see going on. We're going to look in DAPI. We're going to do a very quick snap. And I'm also going to use our hardware autofocus. So that means that it's going to look for the bottom of the plate, and then I've told it the offset that organoid is typically going to be at. It's going to find that each time. So that's what that hardware autofocus is. We call it definite focus. So that's all I'm testing here. So first I'm going to make sure before I go into the guided acquisition, we just want to make sure that everything is set up right, because sometimes plates can be different, right? Everything has a little bit of variability. So I took a snap, and I'm going to go into the analysis tab, because that analysis is what's going to give us those XY coordinates. I'm going to run it to check that it's actually working as expected. It's going to identify the organoid based on my current imaging settings and analysis settings. If it wasn't, I could go to interactive analysis and change anything. If something is different on this plate, maybe it's brighter, maybe it's dimmer, you can still go in and interactively change that before jumping into your automated guided acquisition. But this looks good to me. Found that organoid quick and easy. All I care about is finding the XY, right? Where is that in X and Y? And it's going to do that for every single well, which is also really helpful because sometimes you don't have an organoid in every well, so you're not going to waste time imaging or looking for those. It's going to very quickly scan and find if you do have them. So what you can see here is that I'm going through the guided acquisition, and I'm going to pause really quick. So this is in the Applications tab. This is where you would configure your entire guided acquisition setup. The first thing that you have at the very top there is that 2D low mag screen. So I'm basically telling it which experiment settings to put up. All of that you set up in your acquisition tab the way that you normally would. That's the way that most of us are used to living in the Zen software, is the acquisition tab. So you're going to have those dedicated experiments and the settings in there. It's going to pull from those. I've told it that I want to use the 10x objective to screen here. And I told you it's a 5x equivalent because I'm using a 10x, but I'm zoomed out. That's the beautiful thing about our optics. You can actually zoom out to get the field of view equivalent of a 5x. So I actually have the numerical aperture of a 10, but it's zoomed out to the equivalent field of view I would get on a 5x objective, which is really nice. So I can get that whole well snapped with the 10x objective. So that's where I pick the screening settings. You can see then you have the analysis settings, and that's what I just showed you. I tested again to make sure that it's right? We always want to measure twice, cut once. And so I'm pulling those analysis settings from that analysis. I just double checked that I'm happy with how that is currently set up with my plate. And then the last thing you pick is your actual 3D screen, the snaps that we're going to take, your high mag, your high precision imaging. That's the last experiment setup that I'm going to click. And here I have a 3-color Lightfield 4D track that I had already set up in my acquisition tab. And that's what it's going to pull from after it finds where those organoids are. It's going to go back with the 20x and use those experiment settings. Okay, so we're going to go ahead and hit start. I am going to use a little bit of movie editing magic because I'm sure you guys don't want to sit here, even though it is only 30 minutes to get 3D info for a 384 well plate, but I'm going to put the stopwatch on so that you can see how long it actually would take you if you were using this. Let me move my big old head out of the way. So first thing we're going to see here, I've started the stopwatch, and it will be sped up, but what we're going to see here is it screening the plate with that 10x objective in confocal with the pinhole wide open. We're looking at 𝗶, and we're going to watch it screen this plate, right? So you can see you get the whole well view, and it's just going to very quickly screen through that plate in about 6 minutes or so. And of course, you can, again, tweak all of these settings. I have the hardware autofocus set up to go every 5 wells, but if your plate is flatter than mine is, you can go every 10 wells or something. Every time you cut down a focusing step, that speeds things up. But all this is automated. I'm not clicking anything from here. The next step is going to be actually capturing that 3D acquisition. So it's made that big old analysis table. And now this is super sped up because this took about 30 minutes to get through the rest of the plate after that initial screen. And so it's going through, it's referring to that table where it has the XY coordinates for each individual one that it found at 5x, and it's already switched to the 20x objective, and it's snapping in 3D. We're getting 3D information now, and you're capturing the whole organoid in 3D. I cannot express how exciting this is, and honestly how I wish I had access to this when I was in grad school. I maybe would have gotten out faster, who knows, or gotten more publications. But you can very quickly screen through, and again, 3 colors, 3D, and we're going to be able to segment the individual cells in this to actually get out more reliable data. Okay, we do have the typical trials and tribulations you might have with the plate, right? These are not, this plate that I was given is not fixed in Matrigel or something. These are floating spheroids, but for the most part, these don't move around that much. So that's something that you just is sample dependent. If you need to do a little bit of tweaking on that, you can always change how fast the stage moves and things like that. And at the very end there, I think I'm out of where it can actually focus in the bottom of the wells, but very fast. And the last thing we need to do, I just want to address this because we do need to process, right? Each of those snaps is 37 different images that you need to Lightfield 4D process, but you can just plop them into batch processing and very quickly get out that data that is then ready to port into a 𝗶vis for analysis. You could also technically connect your PCs if you have 2 PCs and do something we call direct processing. It's really whatever workflow works best for your configuration, but the batch processing is so fast here that I honestly just drop it in when I'm done, and then we port it over to the hub, and now we're ready to actually get crunch numbers in under 45 minutes on a 384 well plate. So here we're going to hop into the segmentation and analysis, which again, at the end of the day, is really what we all care about, is getting our data out. So here's the processed image of one of the wells, just to show you an example in a 𝗶vis. I'm working in a 𝗶vis Pro here. And you can see, right, we've got green positive cells, we have red positive cells. What I want to get at the end of the day is a ratio of red and green. You might want to measure the entire volume. You can do that. You can measure the volume of all of your individual segmented cells. You can get really any measurement that you can dream of. You can get it out of a 𝗶vis. pipelines are fully customizable. But for today, I just wanted to see what is my ratio depending on my initial culture treatment conditions and depending on the drug treatment applied. What is the ratio of red to green cells throughout the plate? That's the data that I cared about in this case. So something else you might notice, there are cells around the plate, right? We have cells in the spheroid itself and we have cells around the plate. Normally this can be really like pulling your hair out to try to like not count some of the cells, only count the cells in the organoid. But it's very easy with the machine learning setup that we have in 𝗶 Pro to literally like, it's like coloring like an adult. You can go in and train a machine learning model just by basically coloring in what is positive for a spheroid and what do I not want to count as background. And so that's what we did to basically make a subset of images or a subset of cells only within that spheroid is what we want to count our data for. And so you literally go in and color using the AI tools in 𝗶, and it'll identify this is the object that I care about as a spheroid, this is the background, and you can just train it on that. It really is that simple. And so from there, we were able to port that over into our analysis pipeline and say we only want to count anything that is in an object that we're calling a spheroid. Okay, and I also just want to give big kudos to Jason Fung, who is the specialist for 𝗶 for the western half of the US because he did a lot of this work. But he let me voice this over for him today. So we have green cells, we have red cells, and this is them segmented in 3D, right? We're getting, again, I can't harp on this enough, this is 3D data. We didn't have to sacrifice anything to go fast. We snapped and we got our entire spheroid captured in one shot. And now we can actually read out the population of cancer cells to fibroblasts in this case and get that all out in a big data set. So again, I mentioned that this is fully customizable. Whatever readouts you want to get, you can get, but we're just going to look at this one specific well here. You can see we've got the RFP positive count and the GFP count, and what I said I cared about in this case was the ratio of RFP to GFP. But if you care about something else, you want to normalize all of that to the total cell counts in your organoid, you could do that as well. You can dream it, you could do it. There's any sort of metrics that you want to get out of your data in 𝗶 you can get out. So what we're going to do is, now that we're happy with how it's segmenting the red and the green, we're going to port this over into the 𝗶 hub, which is a really nice tool if you are dealing with giant data sets like this. You basically have your pipelines over there, and then you can drag in the data and then have it just start to chug along for you, right? And so the idea here is you're doing one click and it's going to apply it to everything. So here you can see this is what the navigator looks like in the hub. And so you can see the green checked examples, it's already processed those, and it's going along well by well, right? The nice thing about that guided acquisition, because I drew it over an underlying plate carrier, is it automatically added each well name to each of the names. So you know exactly where you are in the whole map, and when you port that into the hub can also read in that metadata and map it out for you, make a heat map or something useful like that. So let's just look more closely at this well here, the resulting analyzed data. We can click through and look at where are the RFP positive cells, where are the GFP positive cells, just like we were doing in 𝗶 Pro, but now we're in the hub. And we can also go through and actually walk through the Z-stacks of those. So anything, if you wanted to take a closer look before you're looking at your data, or maybe you've already looked at the data, you want to go back and see what did that look like in the segmentation, you can do all of that here. And again, we can go Z-slice by Z-slice and look at the different populations. So super helpful if you just need to go back in and probe and see. Once you can always have your chart of numbers, but maybe you just want to double check that everything looks good to you. You can always do that sanity test back in the software. Again, I want to point out how quickly this is analyzing everything because it's on the hub. It's getting through each of these 3D analyses in like 25 seconds, which is great. So again, we just put it in here and we let it chug through all the data. And at the end of that, we are able to get out a really lovely heat map. So let me switch over to that. All right, so here are our results. We knew we didn't have organoids in every single well, so we do have some blank wells. But you can see all of this data that we got very quickly. We really just clicked one button to do the entire experiment. And again, it gathered all that data in 30 minutes, and then we did a quick processing step, plugged it into our analysis pipeline, boom, you're done. You know, it's very easy. Once you have a robust, if you have like one of those robust, I'm doing this type of experiment every day, day in and day out, multiple times a day, this is exactly the combination of tools that I would want to use if I was still in industry. But I'm trying to help you guys, do science that really is changing the world. And so I'm excited for things like this that come out. So again, we've got a heat map here, and just to give you an idea, I know 384 well plates can be a little bit overwhelming sometimes, or maybe that's just me, but just so you know, this is the treatment conditions that we were given when Inspira gave us this plate, and we can see how that sort of correlates here with this whole heat map in terms of our RFP and GFP ratios. We're seeing, depending on the different culture conditions that were applied, we see that there's a higher ratio of GFP to RFP cells in the left half of the plate, and it does seem like that's also corresponding to different drug treatment conditions. And on the right side of the plate, those were different culture conditions, similar drug treatment paradigm was applied, but we can get all this out, put it out in Excel or whatever you need to then export it into your statistical software and actually start getting some meaningful data from this. All right, so with that, that's really what I wanted to show you guys today. Again, thank you for joining and spending your time with us. We are here to answer your questions, and send us an email. I dropped the email in the chat already. It's also in the information below, but we're here as part of your digital customer engagement team. And I believe we have a few minutes before our next talk will be on Copilot. So that's another really useful tool that's come out. And so come back in a few minutes and we'll tell you how we've built in AI into our microscopy software to really help you guys get to your data as quickly as possible.
Welcome back. Thank you, Rebecca, for that educational presentation. It's my privilege to introduce our next speaker, Amita Gurur. She joins us via Lawrence Berkeley National Labs, where she earned her doctorate in comparative biochemistry. Looking forward to her talk on our Microscopy Copilot. It's an AI assist, and if you have embraced AI to add efficiency to your life as I have, I trust that you'll find this next talk very insightful. Thank you, and away we go.
Hi, everyone. Thank you, Nate, for a wonderful introduction. My name is Amita. I'm coming to you live from the ZEISS Microscopy Customer Center here in Dublin, California. As Nate gave us the introduction, I have the privilege to talk about Microscopy Copilot and all the fun that I've been having with the Copilot. So this is your personal AI assistant. Ask away questions about how to set up your image acquisition on the ZEN software, and also any button that you find you're curious about to learn on ZEN, ask the Copilot, it'll tell you about it. So in the next few minutes, I'm going to walk you through all the fun I have been having with Copilot in a video, and the questions that I've been asking. I really hope you enjoy it, and I really hope that you get to play with it and have as much fun as I did. So let's go. So my talk is optimized imaging with Copilot. AI has completely revolutionized the way we do microscopy. I personally do microscopy in the last couple of years, from image acquisition to analysis to service. So at ZEISS, we have integrated AI into microscopy, and some of the examples are, for example, the AI sample finder. So with this, I have completely stopped using eyepieces. I just put my slide and use the sample finder feature to find my sample. That's how we've been doing microscopy lately. And just to take you through some few other examples of AI, you have image acquisition with guided image acquisition, denoising, image segmentation, object classification, and also predictive service, which will remotely monitor the health of your system. And so here we are. We have launched the Microscopy Copilot, and this is your personal AI-powered assistant. And with this, I will jump right into our microscope. This is our ZEN 3.12 on our LSM 990 and LSM 900 microscope. You log in by clicking on Copilot on the bottom right into the My ZEISS account. This is a free account that takes you to your orders, all the instruments you have in the lab, the learning resources. So this is your one-stop shop for everything My ZEISS in terms of learning resources. You launch into Copilot, and the first question I might ask, it gives you a few prompts. How do I set up a Z-stack? So I asked, and it told me in a very step-by-step instruction, this is how you set up a Z-stack. Check the Z-stack box. You set up the range, top and bottom, and that was very informative because I went about setting a Z-stack with the information it provided me. Here's my beautiful looking 𝗶 elegans sample on a slide. I'm playing through the slices, and then I ask a question. Do I have the LSM Plus module? So if you're curious, if you have license for a module, it actually tells you that. And then I asked, do I have the deconvolution module? It says go to Tools, click on Modules Manager. And I scroll through. I did not find Modules Manager, but I found Toolkit Manager. So I said, hey, Copilot, I see Toolkit Manager. Is that the same as Modules Manager? And it said, aha, you're right, because the ZEN version has upgraded and it's the same, but it allowed me to go in and check if I do have the license. So it's a very interactive way for you to find out even about your own system. And the next question I asked, hey, my image looks grainy. Can you help me out? Gives me a list of instructions on how to improve my image quality. And voila, I had a beautiful looking image just with instructions from the Copilot. And then I ask a question on time series acquisition. And the fun one next is, do I have time for a coffee while I acquire a Lightfield 4D image acquisition? And it was very smart to tell me, well, it depends on your acquisition parameters, the number of time points, so on and so forth. And so I tried to set up an acquisition myself and see if I really have coffee time or not. So here, this is me setting up an acquisition with our latest technology, Lightfield 4D. This is a honeybee leg, and it was so quick that I actually did not have time for coffee post this acquisition. And this is your 3D volume, instant volume, right? This is just a fantastic technology right now within ZEISS Microscopy. And I would encourage all of you to watch our other talks in the workshop. And then finally, I mentioned to Pilot, looks like I don't have time for coffee. And it said, well, if only the microscope can brew the coffee for you. And so the next question I asked was, I'm poking around on Zen, and I find that on the Applications tab, I'm intrigued by this feature called guided acquisition. So I asked the Copilot, what is guided acquisition? So it helps explain in a paragraph what is guided acquisition. And then I ask, all right, can you help me set up an experiment for guided acquisition? And it gives me a step-by-step instruction on how to go about setting up a guided acquisition workflow. Very, very descriptive. So with that, you open up the tab and follow along. And it was easy for me to read those instructions and follow along, set up an overview experiment and a detailed experiment. Here, I'm just highlighting another talk from our workshop. This is high-throughput 3D organoid screening utilizing LF4D. Encourage you to go watch it. And then finally, here's a fun question my colleague asked: how would a starfish wear pants? And there you have those two graphics where you have the starfish with pants and a cape. But these are some fun questions that you can ask the Copilot while you're waiting for image acquisition. So as you could see in my short lightning talk video, you can utilize the Copilot to help you set up image acquisition. How do I do something? So your prompts can be, how do I? So how do I set up a time series? How do I set up a Z-stack? And then while you're waiting for image acquisition, you can be curious about all of the different features on Zen. What does a button do? So on and so forth. So like I said, I've found this extremely useful. And what I see as a really useful feature for Copilot is training, right? So you have folks in the lab who want a refresher from their training session, or just are curious to learn about. It's a self-exploratory experience. So you can ask the Copilot questions and learn along. No better way to spend time in the lab with your instrument. So I wish you all happy imaging and ask away questions with the Copilot, and happy learning. Thank you all for watching, and we'll be right back with the next session.
Welcome back. Thank you, Amita, for that look at the world's first LSM AI assist software. I hope that was engaging as it was for you, as it was for me. And now we're going to move to our next session. It's my privilege to introduce our next session, where our speaker will be speaking about spectral imaging with our next gen LSM. 𝗶 Bjornson has decades of expertise in microscopy, from working as a microscopy core manager to years supporting the MBL community. So thank you for your continued attention, and I hope you enjoy this next session.
I think a good place to start is basically talking about the fact that fluorescence imaging in general is really quite powerful because it allows us to effectively, if things are done right, melt away all of the background and only look at the structures or the proteins or the RNA that we're interested in. And for example, this was done long ago on an LSM 510. And this is it's only 3 labels at this point, but this was astrocytes, blood vessels, and GFAP in a 100 micron thick rat brain section. And you can see that there's really no background at all. It's basically just the proteins that we're looking at, even though the tissue is that thick. And so this really gives us a powerful technique that we can work with to be able to really label everything that we're interested in. And so we've been working with molecular instruments recently to work with their 10-plex HCR gold spectral imaging. And this is a really incredible platform that allows us to basically work with 10 channels at once. So we don't need to strip and then restain with this. We can basically just capture all 10 channels in a single acquisition. And we can do this with either RNA or protein imaging, which is really fantastic. So you don't have to pick and choose between doing FISH or doing antibody staining. It works for both, really. The platform is nucleic acid based, and it amplifies in a quantitative way. And so this gives us a way that we can really quantify signal in tissue sections very nicely. What's even better about this is that working with this group, they've sent us all of the right controls. And so you can see the slide box here. And this has basically every control you could ever possibly want. We have the 10 labeled sample. We have single positive controls for everything, every single one of the labels. And we also have negative controls as well. We have background autofluorescence, and we also have a slide that was prepared with the true black that they're using to quench fluorescent signal as well. And so that is really helpful when we're trying to set up these kinds of acquisitions. We're looking at tonsil tissue in this case, and so we have really a combination of different cell differentiation markers. We have keratin, we have a few different 𝗶I67 for cell division, we have some cell differentiation markers like FOXP3, and that's basically what we're going to generally take a look at today. The emission wavelengths for all of these are highly overlapping, as you can see here. And so this normally might present a problem, right? You would expect crosstalk between these different signals, but with the LSM 980, we're able to basically tease these apart fairly easily. And it's worth mentioning as well, one of the things that I've learned over the years is that these overlapping spectra aren't really the big deal, actually. If we're doing spectral imaging using the quasar detector, or if we're setting very discrete bins as well, we can do. This is usually easy to tease apart. The killer usually is excitation overlap. And so if you have a detection range that captures different emissions, then it only gives you trouble if you're actually cross-exciting those floors in the first place. And so that can be a big deal. And so this is where we're going to change tracks a little bit compared to something that Rebecca Duffy has shown in the past on these different microminutes. And there's a link in the YouTube video below if you want to check that out. It's definitely worth a nod. So we're not going to do single track spectral imaging this time. We're going to end up doing instead multi-track imaging. And so this way we can very discreetly separate different channels and bins, and we can end up exciting with very specific lasers in combination so that we don't end up cross-exciting those fluorophores along the way. In the end, what we're going to get is this. So here, basically, we are looking at a 10-track image, and we are able to basically go into Zen. We're able to add linear unmixing as a step in the direct processing. And so we can automatically unmix at the end of it. We don't have to waste our time going in and doing an extra processing step. We can pick all of the different fluorophores, which you can see populated here. And when we scan this image, you can see that we're going to end up getting a few different fluorophores. They're not, all of the fluorophores aren't necessarily in a particular region. If we did a tile, we could get more, but for the sake of brevity, we're going to do it this way for now. And with this, you can see these coming up in different tracks, and it'll just take a minute for 3 tracks. I'll show you how these are configured in a second so that you have an idea how we built this. But you can see a couple of different signals coming up already, and you can see basically here all of the different channels coming up. We also have a residual channel here as well. The residual channel is a good way that you can actually tell whether or not any of the signals are not fingerprinted well, but I'll show you how we get a fingerprint in a second also. Here's the 3rd track coming up. You can see the 405 channel shows up really nicely in this area as well. And so this is basically in the center of a lymph nodule in the tonsil, and shows up really nicely for some of these CD markers and the 𝗶𝗄 markers. So it'll just take a moment to finish, and we're also doing LSM plus. So it's giving us better resolution than we normally could get with confocal. And now we're going to shift through these different markers here, and you can see very nicely each one of these is well separated from the rest. You can see distinct markers for these HCR probes in each of the different channels here as we move through all of them. Some of them you can tell that they're actually not there because we've got this min-maxed and you end up seeing just background. But really, this is what we're going for. So how did we get here? We ended up setting up a multi-track configuration in this case. We didn't do full spectral. We could have done this in a single pass, but in this case, what we're able to do is set up 3 different tracks. And you can see that we have separate fluorophores on each. And with each one of these, we're setting up a different laser and intensity. We can also set up different gains for each. And the other thing that you'll notice if you take a look on the left-hand side is that for some of these as well, we have, 2 different detector ranges for, let's say, the red and the green here. And what this does is this allows us to tease apart some of the different potential overlapping emission spectra that we might end up having from cross-excitation in this case. And again, here we have a couple of different green tracks, and this allows us basically to really generate spectral fingerprints that are going to work for each one of these different fluorophores, even when they're so highly overlapping. And so just to give you an idea of what one of those might look like, you can see here that we have taken a single positive image, and this is for the 405 channel in this case. Nice strong signal in this region. And so we can work with the unmixing tab. We can use either a small region or we can use a crosshair to pick out a really nice bright individual spot, individual pixel. Oh, thank you. So here, sorry about that, bright spots that we can easily see with the 405 single positive. And we can either use a small region or we can use a crosshair to end up generating a fingerprint. And you can see the unmixed fingerprint here. So very clean signal in the 405, not a lot of emission in the longer wavelengths. And this makes it really easy to work with this channel. And you can see a second one here. This is the 425. So very close spectrally, but this gives us a very nice clean signal with the kind of multi-track configuration that we have set up here. And just to give you an idea of what this looks like, one really good thing to do before you really dive into working with your precious 10 labeled samples is to go ahead and work with your single positive controls. And after you've generated fingerprints for each one of these, really good idea to apply all of the different fingerprints that you've collected to make sure that they are working as you expect. This is your sanity check. It's really important. By doing this, what we can do is make sure that the fingerprint that we collected for one channel and all of the rest of the channels are basically playing well together. And so what you want to see when you do this unmixing is you want to end up seeing the signal from the positive ending up just in the proper channel, and then you don't end up seeing anything else in the rest. And if we do this for each one of the single positives, then this means that we can go ahead with confidence, knowing that the fingerprints we generated are working properly, and that we can apply them to our 10 label sample without trouble. And the other thing to do, there's many different sanity checks, I guess. That's one thing to say. One of the things you can do just for your peace of mind is as you're designing the experiment in the first place, if possible, you can spatially separate any of the fluorophores that might be spectrally close. So if you have 405 and 425, if you can basically place those in different regions of the tissue so that you don't end up wondering whether or not you're getting any kind of bleed through or colocalization. The other thing that you can do is you can do a couple of different controls as well. So one of the things that I've run into is even with highly cross-adsorbed antibodies, you can end up having cross reactivity between those as well. It's not a case with the HCR here, but you can end up seeing that mouse primary antibody might end up getting tagged by a highly cross-absorbed guinea pig secondary antibody, for example. And that ends up factoring into the kind of unmixing that you're going to need to do. An additional control that you might sometimes consider is actually to use one primary with all of the wrong secondaries and just make sure that works. Anyway, here we are. And you can see the individual blue label looked really good. And in the other cases here, we generally don't have signal where that blue stain was. This is all background fluorescence. And so we are good to go with the 405 here in this case. The other thing to mention is that, actually we did that already. How are we doing? One of the things that we did to get here actually was use AI Sample Finder and switch to oil. And so with this, it's a really nice way that we can work with the Zen interactively. I started this with the Zen Connect project and ended up moving from the 5X to the 63X. And you can see doing that here. So very quickly checking to make sure that we're in focus with the 5X. And after that, just basically using the navigation tool that we've created, the Google map that we've created with the AI Sample Finder tool here to move down to the loading position with 5X and then move away from the slide so that the objective front lens is clear and mark a position so that we can come back exactly to the position that we're interested in. After that, we can switch to the 63X, which you'll see in a second. So we're just going to move away here now. And switch to the 63X. At this point, I stepped in to add a drop of oil. And with that, we can basically move again directly to the position that we were at before. So all of the time that we take to generate a AI sample finder image and to create this overview map is preserved. We don't have to take the slide off. We can just leave it there. So that ends up meaning that we can easily track exactly where the positions are that we're looking for. And in this tissue, some of these markers show up in connective tissue, some of them show up in the center of the lymph nodule, some of them are in the cortex. And so it gives us a really easy way that we can navigate after the fact. And you can see here basically coming into focus with the 63X, and we're ready to go. So after doing all of that, I can bring you back to where we started in the first place, which was the 10-plex image here. And again, we can set this up. We can either do this as a processing step separately if you don't want to do it automatically, but we can easily add all of these different settings here and do this on the fly so that when you start your experiment, you're going to end up with an unmixed image at the end. We've kind of done this already, so I can skip past this a little bit, if I can find it. Yep, so again, basically here is the image being collected, and you can see the finished product here, and once it's unmixed, you can see the individual channels as we cycle through as well. Again, this is a really fun project. It's a work in progress, and generating all of these different fingerprints and looking at the distribution of each of these markers across the tissue is a really powerful way to basically pack in a lot of information onto a single slide that might be from a patient sample or a precious sample, special, maybe monkey tissue, human tissue, those kinds of things that are very precious, hard to come by. And that allows us to really make the most of every single section that we end up generating here. I think the other thing that I might mention quickly as well is that, let's skip through some of this. Yeah. So what we do here, we have proper controls to begin. We have single positives, and we also have a single and unstained controls also. Another one that we could do is single primary antibody with all the wrong secondaries if you're worried about that kind of crosstalk. Spectrally separate labels that are spatially close. This makes sure that you are confident in the unmixing that you're doing. Another thing that I didn't mention is that you can also place brighter signal on off-peak excitation or narrow detection ranges. So if you have the ability to do this up front, then you can make sure that the fingerprints you're generating for each one of the spectra are generally kind of uniform in intensity. And that's going to make sure that you can have signals that don't really rob from each other as you're working with them together. Another thing you can do is take channels that suffer from low signal to noise and put them on separate tracks so that they're not being overwhelmed by other channels as well. So a few things that I've learned over the years, and I hope that they can help other people as they're sort of beginning their multiplexing journey.
We're back in the studio room live from the ZEISS Microscopy Customer Demo Center in the Bay Area. I hope you found that presentation from Chris enlightening and speaking to how we can entangle the complexities of overlapping dyes. We'll be right back in a few minutes. Thanks for tuning in. And as a reminder, if you have any questions, you can contact us via the email address in the upper right-hand corner of the screen. We'll be back in just a few minutes. Thank you. Welcome back from that short break. I hope you enjoyed Chris's overview and introduction to spectral unmixing with our next-gen LSM. And now it's my privilege to introduce our next speaker and session, a continuation of spectral imaging relative to using linear unmixing to look in tissue for background subtraction and addressing autofluorescence challenges. Our next speaker, ZEISS, joins the Digital Customer Engagement Center team after several years in academia, jumping from the University of Florida to the University of Minnesota, earning his doctorate in biomedical engineering. So I'm going to switch it over to ZEISS now. Thank you and enjoy.
Hi, everyone. In this session, we'll be covering how to remove background from your samples using linear unmixing. So on the microscope right now, on the LSM 990, I've got this slide with a mouse brain tissue, which has been labeled for GFP, cytoplasmic GFP for the neurons, and then DRAC5 labeling all the nuclei in the tissue. So as you can see in this overview image that I've taken here using a 10x objective, and if I zoom out, you can see the actual slide, you see that the whole tissue is fairly green, indicating there's a lot of background autofluorescence. And if I zoom in onto the hippocampal region area over here, you can see some of the neurons in the hippocampal region. And so everything else we see out here, that's autofluorescence. And unfortunately, the autofluorescence is green, same as the GFP, so it shows up in that channel. So in the next 10 minutes, I'll be going through how we can remove the autofluorescence using linear unmixing. So now as I switch to our navigator view, you can see the tissue. I'm using a 20x air lens right now. So in this little square blue box is where my current position is. So I can drag that around to navigate. So what I'll do is bring up a live image. And right now, because I have GFP cytoplasmic and DRAC5, I have both channels simultaneously using the 488 and 639 laser lines. And if I bring up a live image and zoom in onto my navigator view over here, I can see in the GFP channel, the cell body and the dendrites over this cell I was looking at. And then if I turn on the DRAC5, I can see all of the nuclei within this image. So from this image, you can tell that there's quite a bit of background. So if I snap this image right here, you can see from the image, if I do the split view, on the left-hand side, we can see the cell body and the dendrites, but we have quite a bit of background here. And then within the DRAC5 channel, we can see the nuclei, but then we also have quite a bit of background. So outside of, redoing the sample prep, what can we do? And this is where the linear unmixing can come in handy. So now instead of doing confocal imaging, I'm going to turn on our lambda imaging track. So with the confocal track, you can see here I have two channels, GFP and DRAC5. But when I go to the lambda track, you can see I can use my spectral detector, which detects the full range from 375 nanometers to 900 nanometers. And I've got my 488 laser on right here and the 639 laser on. And let's see what we get when we use the spectral detector. So now when I bring up a live image, what you will see is here we're seeing the true color. So you can see the green cell body and the dendrite. You can see some of the red in the DRAC5, and then this speckly green, yellow, orange, red. That's all of our background autofluorescence. So here you can see we're actually acquiring 32 plus 4, 36 channels at once. So we've got this channel S, which is the 32 channel array, channel 1, channel 2, and the two NIR external detectors that are additional 4. So we're acquiring 36 channels simultaneously. So when I snap that image, we're seeing the true color, where previously we were looking at pseudo color with these two channels. I can pseudo color these any color I'd like. So I've labeled them green and magenta to be colorblind friendly. So now when we go back to our lambda image, if I look at the split view, you'll see every single channel. So within this, I can actually add an annotation here so that we can look at the wavelength that we're looking at. So if I click, I believe it should be under Show Dimensions Annotation. Let's make that text a little bit bigger. Oh, it's a little bit too big. So we can see precisely from about 400 nanometers to 900 nanometers, we have 36 channels. So if we zoom in into this 500 nanometer range, we can actually see that's where the cell body is, but you can see it has some autofluorescence going out into the yellow-orangish region, and then in around the 750 nanometers in the far red, we can see the DRAC5 signals. Now if I go into the unmixing tab, let's see what signatures we've got in this image. So by taking a region of the image, let's say this rectangular box, I can actually see what the true fluorescence is there. So you can see there's a peak at 550, which is green, and then there's a peak around 750 to 800, which is far red. And if I move that around, if I move this over the DRAC5, you can see how that shape changes. So now over here, I've got more far red, less GFP. If I bring this box over to the cell body, I have more GFP, less of the DRAC5 and the autofluorescence. So by using this, I can actually see the exact fluorescence profile, the emission spectra I'm getting from my samples. So if you know that we've got two labels in this, which I do, and you have background autofluorescence, we can actually use the software to identify all those three species. So if I identify three species, one is the GFP, one is the DRAC5, one is the autofluorescence, let's see what we get. Now we've got three curves. So if we look at those three curves and look at the x's, we've got a blue X over the cell body, we've got a red X over one of the nuclei, and then we've got a green X somewhere where the autofluorescence is. So let me change these colors. I'm going to change the first signature to GFP because I know that's my cell body with the cytoplasmic GFP. The second one I'm going to make, let's do light blue. That's going to be my background autofluorescence, or BG. And then over here, my red, I'll turn into violet or magenta, and that's my DRAC5. So once I have these spectra, you can see if you look at the theoretical emission spectrum of GFP, this should match what we see when we look it up on FP-base. Similarly with DRAC5, if we look at a spectra viewer, we should see it matches. And interestingly enough, you can see the background signature profile is very green, slightly shifted red from GFP. It's also got autofluorescence in the far red. So it's not surprising that over here we had a lot of autofluorescence and background in both the green and DRAC5 channel, because we can see from the spectra that we pulled from our lambda scan that the autofluorescence is green and red. So if I click this linear unmixing, we can then see the result. Now we get 5 images when we, or not 5, sorry, 4. And if we look at the split view, this is what we get. We've got one channel with the green with hardly any background autofluorescence. This is our background autofluorescence channel in the blue. And then we've got the DRAC5, which has barely any autofluorescence. We also have this residual channel. So when we do the linear unmixing, we're using some fitting to the data, and the residual channel is showing us the error of the fit. So the residual channel should be a noisy image. It shouldn't look like any particular structure. So you can see the signals are very dim and it's just a fairly noisy image, which tells us this was a good unmixing result. If in the residuals channel you saw some structures, like the nuclei or the cell body or the dendrites, then you know it's reassigning those photons from these 3 channels into the wrong place where the residuals would be. So we have a bad fit. So we would have more error in that fit and higher residuals. So now that I've confirmed that this is a good fit on the first pass, I can go back to my image and actually save these fluorescence profiles so that when I image this slide in the future, I can use this. And I no longer need the residual channel as well because I know the error is low, the residuals are low, so I can throw away this channel. So now I can save these 3 dyes to our Spectra database, and I'll save it as brain slice under that folder, and then within this folder, I'll save GFP, and I'll save the background, and then I'll save the DRAC5. So now, if I were to take another lambda stack, let's go back to my navigator and go to a different region, maybe over here. We've got a lot more nuclei over there. And over in this region, if I hit snap, I'll get a new lambda stack. And then from here, I can go into the unmixing tab and load the spectra that I had identified previously. GFP, background, and the DRAC5. I don't need the residual channel anymore, and click unmixing. Then I should have my 3 channels, one with the GFP, one with the background, and one with the DRAC5. And now we can actually even see the dendrites on this neuron, where previously it was hidden amongst the background and the DRAC5 when we did the lambda scan. So now, obviously, if you want to take a Z-stack or tiling and so on, you don't want to have to process each image individually. So now I'll show you how to create a online fingerprinting, which is where we're going to do this unmixing on the fly. So now I'm creating an online fingerprinting track, and I'm going to include my brain slice folder. So it's going to include the background, the DRAC5, and GFP, and I'm going to create this track. And I can scan all 3 channels simultaneously by performing a lambda scan live and doing the unmixing live. So we call that online fingerprinting. So now if I go to my navigator, move to any region within this tissue, and start a live scan, we should see just 3 channels. We'll want to turn on the lasers as well. So I had the 488 at 1% laser, and then we had the DRAC5 at 0.2% laser. Now that we have the lasers on, we can see in the split view just the 2 clean channels by themselves. So now we're doing the lambda scan and unmixing on the fly without any need to post-process. So now if I were to take a Z-stack, I'll turn on my Z-stack right here, take maybe a region slightly above this, and then take another region lower, down here. So that's about 10 microns. And if I sample every 1 micron, that's 10 slices. And hit start experiment. So now we will be getting a Z-stack where we can separate out the background autofluorescence from the sample. We've got the DRAC5 nuclei cleanly separated without any background, as well as the GFP, cytoplasmic GFP, labeling the neurons with hardly any background. And once this Z-stack finishes, there's one more trick we can show you, which is that because we know, you may not need to visualize the background, we can actually uncheck that background so we don't even have to acquire it. We'll only acquire the GFP and DRAC5 and throw away the background so that we can save some file size so that we're not acquiring 3 channels and only acquiring 2 channels. Here we go. So if I turn off the background channel and hit continuous, you can see that here now I'm only acquiring 2 channels instead of 3 channels. And we've got the DRAC5 and then GFP over here. So just to wrap up, what we started in this session was we started with this brain tissue slide over here, and we took 10x overview we took an overview with the 10x magnification, which I had already done prior. We imaged a couple spots, and you can see within this project workspace, I can actually even see where I've imaged. And when we look at the traditional confocal scanning with the 2 channels, you can see quite a bit of background in the GFP and DRAC5. But by doing the unmixing, we were able to separate the background autofluorescence. And then after that, we moved into doing online fingerprinting, where we combined everything we had done in this session and did the lambda scan and unmixing on the fly so that we didn't have to do any post-processing. Thank you for your time and hope you enjoy the rest of the sessions. Thank you everyone for your attention. I hope that was educational. We've got one more session for the day. I'm going to be bringing back Amita Gurur, who will be talking about an end-to-end workflow for spatial biology. And again, if you have any questions on what you just saw in the previous session, feel free to email us over here.
Hi, everyone. So this is Amita back live from the ZEISS Microscopy Customer Center. So you're attending the last session of the day, and the topic is one-click slide scanning and analysis for spatial biology. So this is ZEISS's offering utilizing the ZEISS AxioScan 7. And my goal today is to show you how with one single click, you can analyze 100 slides in a run from scan to an upload to the server, followed up with analysis. So spatial biology is a field that has applications in many different fields. So in this example, we have immuno-oncology, infectious diseases, inflammation, metabolic diseases, neurology, etc. But just taking an example from immuno-oncology, a system like the AxioScan, so if you have slides that are well stained and prepared, it can image many different slides, up to 100 per run, that have been stained by multiplex immunofluorescence. And after loading on the AxioScan, it can give you subcellular resolution and followed by analysis, which will do cell segmentation, phenotyping, followed by spatial analysis. So this is pretty much going end to end from starting with a slide, scanning, and analysis at the end. So let's review some of the challenges in routine spatial biology. Some of the challenges are you have very complicated multi-immunofluorescence staining. The process is cumbersome and manual. there is very limited throughput, the results are inconsistent, and oftentimes researchers are faced with very complex data analysis. So to address all these challenges, the solutions that we have come up is, what if we provided you pre-optimized reagent kits? So these kits currently we offer from two companies, the UltiView and the 𝗏𝗏, and there is seamless automation for imaging all of these slides from image to results. And the instruments, there is consistency from one instrument to the other in terms of image quality as well. The last point is, all of this comes with pre-trained image analysis routine, so the user does not have to have, background in image analysis, because these come with pre-trained models as well. So our end-to-end spatial biology workflow starts with the imaging kit itself. And these, as I mentioned earlier, come from UltiView. So the offering comes with a kit, staining kit from UltiView, and with consultation on staining as well. Once the staining has been done by you with the slides, you can get them loaded on the Axioscan. And I will show you in a little bit a workflow video where we utilize the slide stream software, which is a very user-friendly, just a single click operation. And once the data has been collected, it is automatically uploaded onto the server for analysis. So essentially, this is our end-to-end offering, starting with providing you a staining kit, imaging on the Axioscan with the slide stream software, automatic upload, and MindPeak is the analysis software that we utilize for image analysis. As I mentioned, the two kits are from UltiView. This is the 5-plex, or the kit is very well optimized, so there are detailed protocols on staining. And the other one is 𝗏𝗏, which offers up to 8-plex and is imaged with a specific filter set that is available on the spatial biology Axioscan that can do spatial separation for up to 8 channels. So this is an example of all of the 8 channels imaging with the streptoclick methodology offered by the 𝗏𝗏 kit on the Axioscan. Let's focus on the imaging aspect on the Axioscan. I will toggle very quickly to the workflow video, and so you can see how this is done. All right. So in the next couple of minutes, I'm going to walk you through the slide stream software that comes loaded on the Axioscan 7, and all with one single click. So I have loaded the Axioscan with 2 slides that have been stained with the 𝗏𝗏 kit and the UltiView. But of course, you can also load all the 25 trays with 4 slides each, which is up to 100 slides. And with one single click that I point on the video, you can start the entire workflow that goes from previewing the slide all the way to upload. But let's follow that video and see how the first step, where my slides had a barcode, so there is automatic retrieval of all of the information that has been encoded in the barcode. So you can tell what is the sample name, the label, what assay was used to image, what was the profile. So all of the scan profiles are pre-configured and optimized and loaded on the Axioscan, as I mentioned on the video. So, and what are the different channels that are being imaged? So if it's a 4+1, or 5-plex from the UltiView, 𝗶𝗶, 𝗫3, 𝗫5, 𝗫7, 𝗏𝐻𝑇se. So all of this information is automatically identified as soon as it starts previewing the slide. So that barcode gives you that information. And as you see on the video, it goes from sample recognition to scanning to what you see right now is uploading. So the boxed view of the in the video shows you the different steps along the workflow, starting from, like I said, preview, sample detection, which is automated using AI models, and now upload, which is straight to the server for image analysis on the with the MindPeak. And at this point, it's repeating the same steps for slide 2 on the same tray. It goes through the same steps. It's almost like a pizza tracker, like I like to say it, which is on the top. You can click on the button up to which you want to run the workflow, but it's literally just one single button which says run workflow. So you click on that button and it goes all the way from start to finish. And once it's done, you can see the word done. So you know that the upload is complete. And so when you open the image, it automatically launches you into the MindPeak server. So essentially, the integration between the software is sleek and very seamless. So what you saw me doing there is clicking on the globe icon in the SlideStream software, and it automatically launches into MindPeak. And all of the different functionalities on MindPeak, I'm turning on individual channels, and then it helps me toggle between different channels on and off. And so here I'm visualizing an 8-channel slide stained with the Chromnigon kit. And once I am happy with the image quality, I can either accept or deny. And once I do that, you click on Start Analysis. So that was your single click for analysis. We did the single click on SlideStream, and now here's the single click on analysis on MindPeak. And once the analysis is complete, so for this particular slide, it took me 5 minutes, and I have sped it up for the purpose of the talk today. Once the image analysis is complete, you can toggle on the MindPeak user interface to check the tumor cells versus stroma. You can get the individual markers, the density, abundance. So what you see me doing on the video is toggling through different phenotypic markers on the MindPeak interface. And you can draw ROI. So essentially, analysis is as easy as right-click and draw an ROI. You can name all of those individual ROIs by different regions as well. And what stands out for me is the rendering is almost instantaneous. And the fact that all these AI models are pre-trained and loaded and ready to go. And this information on image analysis comes from the barcode as well. So the barcode, of course, you can also use text recognition on SlideStream as well. And now what you see me doing is I'm creating a report, and the report tells me all of the information that I can derive from the image analysis. So what is the percentage non-tumor, density, abundance, so on and so forth. And so that was the one-click video that I just played. Essentially, just to recap, what you see on the screen is the interface for the SlideStream software. And on the top right, where it says Run Workflow, that's pretty much the only single button that I need to click after selecting all of my slides. It could be one slide, it could be 100 slides. But essentially, all you're doing as the user is you're clicking, you're selecting the slides, and clicking on that Run Workflow, one single button, which is essentially combining multi-steps. And just to reiterate all of the multi-steps that go behind that one button which says Run Workflow, it's previewing the slide, which is essentially giving the image of the label, but it's reading the barcode, or if you have text or OCR, optical character recognition feature, that's also on the slide stream. It's literally trying to get all that information which is encoded in the barcode, and it does automatic sample detection, which is based on AI models as well. So there's a lot of hands-off automation in this workflow because the user is not manually drawing any of the tissue. So what you saw in that video is pretty much a very hands-off operation because it's pulling all the information from barcode. It is previewing the slides. It's automatically detecting the sample tissue. It is scanning, and scanning happens with pre-configured scan profiles, which have been optimized with the 20x objective. And then it goes on to automatically uploading onto the server and image analysis utilizing MindPeak. So all of that was done just with that single click. So if there's anything that I would want you to walk away from this talk, it's the seamless integration of multi-steps into one button, which is run workflow on the SlideStream software. So I'm toggling back to my deck. Moving forward, I just wanted to mention a few other points on the spatial biology Axioscan. A lot of effort has gone behind automating this instrument and the workflow. So if you are somebody who has used a slide scanner, but you have struggled with tissue detection, what has been perfected in this workflow is the automatic tissue detection. So thousands and thousands, so almost 3,000 plus preview images have been loaded and pre-trained for detection. So especially tissues which are very faint and hard to see, they get detected pretty well. So up to 90 to 95% detection. And so that's AI-based tissue detection. So that comes with models that have been preloaded on the system and ready to go. And few other features that I will walk you through is the feature of high dynamic range. So you may be familiar with samples that have, you go through multiple rounds of staining, and then there are regions that are very bright, and then there are regions that are very dim. So setting exposure time is very challenging, right? So we are taking a lot of time trying to set the exposure time, and then so you have regions that are overexpressed and underexpressed. So what the HDR feature, the high dynamic range feature, gives us here is that the dynamic range is expanded. So what that gives me is that there is even exposure or distribution when I see the histogram. So you don't see that very bright and very dim what you see in the panel above, right? The saturated pixels, you don't see that with the HDR feature. Another feature that we have on the system is fluorescence intensity normalization. So you may have also encountered, if you have multiple units in a facility, the image that comes from one system may not be exactly similar with images coming from another system. So especially if you're in biopharma, biotech, you have multiple units, right? You have a farm of multiple units, and you want reproducibility from one system to the other. So what we have done is we have come up with this calibration, which is done in the factory, and we call it fluorescence intensity normalization. So essentially, if you have then systems in many different locations, all the images come out looking exactly the same. And that is done by a calibration that is done in the factory. And Another feature that is really exciting is cyclic staining and co-registration. So, if you're doing multiple rounds of staining, and then you want all those images to be the images coming from every round to be co-registered, we have a module that does co-registration, and we have seen pixel perfect registration based on this up to 10 rounds of imaging. So this is available on our Zen module, which is our software, and also on the SlideStream software as well. So with that, I just wanted to recap our end-to-end workflow offering, where as a user, you have access to a staining kit, which is very well optimized for the staining protocol and the consultation that you would receive for staining. Once the slides have been stained with those kits, you could then be imaging all those slides on the AxioScan, and the features of the AxioScan, apart from it being a high throughput instrument where you load the slides with minimal training required, because if you recap to the video that I showed, all of the optimization is done in the factory and when the instrument is installed. So as a user, essentially, you're loading up the slides, you're launching SlideStream, which is the software to run the scans, and all you're doing is selecting your slides that you want to scan and start run workflow. And pretty much everything else is optimized in terms of sample detection based on AI models, the scanning protocol. There is a lot that goes behind the scanning protocol, and you can go and check the steps, but the seamless integration on SlideStream is such that all of that complexity is hidden. So the user does not need to really go and optimize any of the scan profiles. It's all of the imaging parameters is all configured and preloaded based on the slide and the barcode. And like I said, from staining to imaging to analysis, because there is automatic upload. Once the scanning is complete, it automatically uploads to the server, and that's where you can use the MindPeak software to analyze your data using pre-trained analysis routine. So with that, that was a quick sneak peek into our end-to-end workflow from complexity to clarity. This is our workflow automation. Years of hard work that have gone behind this in trying to make it super simple in making your research in spatial biology seamless. So hope you enjoyed it. And with this, we have come to the end of our session. Thank you all for joining day one of our live virtual workshop, live from the ZEISS Microscopy Customer Center here in Bay Area, California. And we really look forward to having you all tomorrow, which is day 2 of our live workshop. Many, many more exciting workshops coming up. Please do join us again. And if you have any questions, please reach out to us on the email. Thank you again, and see you tomorrow. Bye.